Effects of potentially toxic elements on soil microbial activity. Potentially toxic elements are common and important refractory pollutants, affect the number, diversity and microbial activity of soil microorganisms. As soil microorganisms decompose organic matter, affects nutrient cycling, microbes play a major role in soil fertility and primary production. Soil microbial biomass, which is useful for studying the harmful effects of toxic metals at the cellular level, is mainly made up of bacteria and fungi. The toxic effect of potentially toxic elements depends on the number of metals bioaccumulated by absorption, migration, and transformation. Adverse effects of higher concentration of toxic metals on microorganisms may be due to inactivation of enzyme activity center, electron-donating groups such as mercaptoprotein, nucleic acid base and phosphate combination, accumulation of toxic metals more than the ability of organisms to bear, resulting in biological disease and death, inhibition in the formation of metallothionine or metalloprotein. A number of studies have shown that the higher number of metallothionine in cells induces the anti-apoptotic effects, and a decrease in the number of metallothionine increases the susceptibility to apoptotic cell death. Soil microbes are the main participant of all the soil biochemical processes. Soil biochemical processes are the tools for maintaining soil quality, formation of soil organic matter, decomposition of harmful substances, formation of soil structure and biochemical cycles. Contamination of soils by toxic metals decreases soil microbial properties such as soil respiration, enzymatic activities. Soil microbial properties depend on soil pH, organic matter and other chemical properties. Severe potentially toxic element pollution can inhibit soil microbial activity and seriously threaten the soil ecosystem function. A number of workers have reported that potentially toxic elements, particularly cadmium, copper, and zinc, can disrupt the microbiological equilibrium of soil. Disturbances of the biological balance of soil caused by the excess of potentially toxic elements might be attributed to the disruption of physiological functions, denaturation of proteins and destruction of cellular membranes of soil microorganisms. Potentially toxic elements immobilize soil bacteria, while microbial metabolites enhance the mobility of potentially toxic elements. Potentially toxic elements in different quantities and forms in soils cause changes in the counts of microorganisms, microbial biomass and microbial activity via inhibiting microbial community diversity, particularly that of fungal groups i.e., Ascomycota and Chytridiomycota, and the large size fractions, which mainly depends on heterogeneous soak availability across the PSFs. Potentially toxic elements create abiotic stresses by inducing disorders in the metabolism of the microorganism. Damages to the control systems regulated by regulatory and signal proteins, including the cell's development, apoptosis and regulation of the cellular cycle, are caused by potentially toxic elements, which might be due to the blocking of enzymatic active centers and driving away cations that are important for the functioning of a cell, supplanting their functions, for example, discontinuation of the cell-to-cell adhesion, cadmium, direct binding with the DNA, chromium, interacting with the binding sites of protein phosphatases, vanadium. Tolerant species of microbes demonstrate higher resistance to stress factors than sensitive ones. Tolerance of potentially toxic elements is associated with 1. Specific transport of metal ions in the cytoplasmic membrane. 2. Synthesis and excretion to the environment chelating compounds, which bind and transport ions dissolved in the environment. 3. Sorption of ions onto mucosal surfaces and the binding by biopolymers of the wall and membrane complex. 4. The presence of plasmids in a bacterial cell, which enables it to acquire resistance to toxic elements. Many researchers have reported that when potentially toxic elements are present in the excessive amount in the soil, the microbial count and diversity of microorganisms decreases. Bansal and Mishra reported that there was a significant increase in the bacterial and fungal population and decrease in actinomycetes population in sewage-irrigated soils. The population density of bacteria and fungi increased with duration of sewage irrigation up to 14 days of incubation and thereafter decreased. Cauchu during their studies found that potentially toxic elements contamination in alkaline soils has a negative effect on actinomycetes and fungi soil populations, while a positive effect on the total aerobic heterotrophic bacterial population. They also infer that the effect of potentially toxic elements on microbial population of the soil is dependent on several factors related to soil environment and soil physicochemical characteristics. Finquayu et al. During their studies on microbial diversity and community structure in agricultural soils suffering from 4 years of Pb contamination found that the presence of Pb2+ in soil showed the weak impact on the diversity of soil bacterial community. Contamination of soil by Pb influences soil chemical properties and number of some genera of bacteria. The number of heavy metal resistant bacteria at genus level viz. Bacillus, Streptococcus, and Arthropteris highly correlated with the amount of Pb. There was a negative correlation between soil organic matter and available Pb and total Pb. The total relative abundance of gemmatiminates, nitrospirae, and planktomycetes was negatively correlated with total Pb. Both the microbial community composition and physicochemical properties of soil are influenced by the amount of Pb. Workers found that nitrifying bacteria, symbiotic nitrogen-fixing bacteria, and azotobacters are the microorganisms most susceptible to potentially toxic elements. Potentially toxic elements produce a stronger effect on azotobacter cells than organotrophic bacteria, mainly because richer communities of microbes are more resistant to potentially toxic elements than single species and genera. Wyszkowska et al. Reported that the inhibitory effect of potentially toxic elements on soil microorganisms can be represented as follows: oligotrophic bacteria, Ni greater than Pb greater than Cr3, greater than Chu greater than Zn greater than Cd, copiotrophic bacteria, Cd greater than Ni greater than Cr3, greater than Zn greater than Chu, ammonifying bacteria, Ni greater than Pb greater than Cr3, greater than Cd greater than Zn greater than Hg, nitrogen immobilizing bacteria, Zn greater than Cr3, greater than Hg greater than Chu, actinomycetes, Chu greater than Cr3, greater than Ni greater than Zn greater than Pb. Few researchers found that crops can moderate the influence of potentially toxic elements on soil microbes. Crops improve the microbiological activity of the soil, mainly owing to substances secreted by roots.